everyone. So today we will be seeing the evolutionary odyssey of human dentition from Australopithecus to Homo sapiens. Beginning with the introduction to dental anthropology. This dental anthropology is an interdisciplinary science. It combines paleoanthropology, bioarchaeology, genetics, and forensic odontology. It studies dental morphology, microstructure, pathology, and variation across species and population. So, dental anthropology, white teeth. So, it has high preservation. Enamel is 96% mineralized. So, it resists decomposition and it is better than the bone itself. Dental calculus, it preserves ancient DNA, proteins, and pathogens, and that is why they chose teeth than bone. The biological archives are still available, and we can see the incremental growth lines, that is a pericymata, which records the developmental stresses, and we have isotopic signatures which are also available in the teeth, like which is seen through carbon dating and all. It reveals the lifetime dietary patterns of the evolution, beginning from the Australopithecus till today's date Homo sapiens. The evolutionary dental metrics, the enamel thickness of Australopithecus was 2.5 to 3 mm thick. It had a thicker enamel compared to current day Homo sapiens. And the adaptation is durophagy. It is since they feed on hard object feeding because they were in a forest-like savanna environment. But in today's date, our enamel is like around 1 to 1.5 ml in thickness. It is due to processed or it is due to cooked, well-processed food. And exceptions, hyperthick enamel in Paleobosis boise, it is before 300 years, and the enamel thickness was up to 3.5 mm. There is also a evident molar size reduction when we look back at the evolution. So, Afransis, it had a molar area of approximately 460 millimeters square. Homo erectus, it had 320, which is evident decrease. And now we have modern humans, Homo sapiens, where our molar size is only around 250 millimeters square. So there is mostly 50% reduction in the molar size, and the causes, it is again due to the softened foods which requires less chewing, and it is also the tool usage. The tools usage, it reduces our masticated demand, due to which the surface area and the molar size is evidently decreased. Next, the cusp pattern means there was this Y5 molar pattern which was seen in apes and early hominins, including Australopithecus. So it is found in Ardipithecus and early Australopithecus species. Y5 molar pattern, it has 5 cusps with Y-shaped fissures. Now in present day we have +4 pattern, that is 4 cusps with cruciform fissures. So it started emerging from the Homo habilis family, that is approximately 2.1 MYA is million years ago. So it started emerging from 2.1 million years ago, and currently we mostly have the 4 cusp pattern. Moving on to the hominin dental adaptations. Australopithecus. So we will be discussing about the dental adaptations on the evolution. So Australopithecus, that is 4 to 2 million years ago, they had meganodontia, that is larger molars relative to body size, and they also had small canines, as canines used for curing food, reduced sexual dimorphism, this is apes. The dietary evidence, they had microbial, that is pitted enamel, because they had hard foods like seeds and nuts. And the isotopes, isotope revelation, it said that C4 plant consumption was there. That is, they consumed the grasses and the sedges, and they used to have a nut-based diet, due to which they had megadontia. And we have early Homo sapiens, which is 2.5 years to 1.5 million years ago. They had a parabolic dental arcade, that is, but the Australopithecus, they had a U-shaped dental arcade, but here it is a parabolic dental arcade. also had a reduced premolar complexity. The honing, there was loss of this honing complex. And the tool-use correlation, this early Homo sapiens, they were correlated with the older one tools, which were used around approximately 2.6 million years ago, and there less need for heavy mastigation. Like they started inventing tools due to which the masticatory needs started decreasing. So this was 2.5 to 1.5 million years ago. And then we have Homo neanderthals, and they have taurodontism. So they have enlarged skull cavities in molars, and there was a very evident anterior tooth wear, that is labial scratches were seen because they used teeth as tools to bite and chew food. And carbon dating, it revealed that the levels, the delta 15 N levels, it revealed hypercarnivorous diet. That is, they had 80% animal food. So there was a very big change from Australopithecus to Neanderthals, because Australopithecus, they had most of plant-based nut foods, and it was hard. And now they have shifted to meat, animal diet. And the tooth calculus, it also reveals cooked plants. They started cooking plants. Previously, they used to have they used to not cook food, but after this early Homo sapiens, they started cooking food, and that was seen in the tooth calculus also. Example, we have this evidence for a research which says that Chandarkir, I will show you the study later. And we have modern humans, Homo sapiens. So it is, we have malocclusion, which is 30% prevalent, and previously it was just lesser than 5% in the Paleolithic population, because the arch shape was wider. But now, because of evolution, our arches have become narrow, and our teeth, the surface area has become small, and now we have malocclusion, and we also have an increase in Gary spike. That was lesser than 1% in Paleolithic era and greater than 20% in Neolithic era. Now it is like there's a very huge Caddy spike which is evident, and yes, the evolutionary mismatch. That is, jaw size reduction outpaces tooth size reduction. So we encounter impacted teeth. Like nowadays, like compared to the previous generations, we face a lot of impaction issues, and it is because of the evolution. And so to study dental anthropology, this evolution, they used these methods. Firstly, they used microscopic techniques like scanning electron microscope. It resolved submicron microwear features, and they also used something called synchrotron imaging to study the Van Ebner's lines. And then we had biochemical analysis using stable isotope analysis, and that indicated that the early species, they had this plant consumption, and now there was a transition from herbivore to carnivore stages. And biochemical analysis, it also reconstructed pulp chips, so that they took the ancient DNA and studied it in some researchers. And we have computational approaches like geometrics, morphometrics, and machine learning, which has been recently used in the current evolution-based studies. And we also have clinical and forensic applications. Forensic identification like dental age estimation using Dentition method, tooth calcification stages will be studied, and Gustafson's criteria, which age from root translucent will be studied. And so this can also be used in mass disasters. So since it has 99.8% accuracy compared to fingerprints, which only has 92% accuracy. And we have something called evolutionary dentistry. So as said, the mismatch theory, modern jaws have become smaller, 15% smaller than the previous species. And this paleodiet studies inform that there is optimal occlusal loading, but still there is reduction in the surface area and the jaw size. So future directions, this can be used in studying tooth regeneration using scab stem cells for bioengineered molars, and paleoproteomics, amylogenin sequencing for sex determination, and indigenous dental remains should also be taken. To conclude, dental anthropology, it is not about the past. It is a lens to understand the present day oral health challenges and future human evolution. As we advance bioengineering, that is stem cell growth, teeth, and precise medicine, our fossilized dental legacy reminds us that evolution, it never stops. And I'll show you the pictures. So, as said, these were the facial reconstructions of what the early hominins were. This is Australopithecus, and this is how it looked like. So this was from the Labrador here. So it was all taken from the Africa, the Ethiopia. This was the location. And so this is Lucy. Lucy is Australopithecus. So it is from the family Australopithecus afarensis. It's a female. It was found at Hadar, that is Africa, Ethiopia, and this was approximately around before 3.18 million years ago. And this is how our present day teeth is. And so this is a study which was taken from Journal of Human Evolution, and this study was about the 3D enamel thickness in Neanderthal and modern human permanent canines. They compared Neanderthal population with canine, our present day canines, and this is red color. Okay, upper canines. The thickest enamel corresponds to red, and the thinness, it corresponds to violet. So it is said that the evolution, because of evolution, we've got thinner enamels. So this was evident from this study. And there is an inverse relationship between enamel thickness and tool use sophistication. So it is a direct imprint of Paleolithic dietary revolution. Similarly, Neanderthal teeth, the ones in the previous era, their teeth were compared to Swiss Army knives because they were used for clamping hides, evidenced by unique layer wear, which is not now seen in present day humans because of the well-cooked food. And this is another study on the evolution of the morphology and resolution of molar cusps in fossil hominid teeth. And it states that the enamel thickness of the teeth and the width in hominids, it promoted the tooth resilience and in turn the lifespan of the tooth, which was not seen before. So similarly, the study also suggested that the occlusal force, which was seen in hominids, it provided the upper bound with the maximum bite force. Because of the higher surface area, they had increased bite force, which is currently not seen. And this is just evolution from the chimpanzee